This video will highlight a simplistic diagram of a process referred to as protein synthesis. First, let's establish that protein synthesis takes place in the cytoplasm of a given cell. But in order to make protein, we need DNA, which is housed in the nucleus. The first part, transcription, takes place in the nucleus, while translation occurs in the cytoplasm. We again call the first part of this process transcription. Transcription can be broken down into three unique stages: 1. Initiation, 2. Elongation, and 3. Termination. For initiation, we have what we call the promoter, a specific set of nucleotides on DNA that, just like the name, initiate the process of transcription. For our example, the promoter sequence is ATG, adenine, thymine, and guanine. This is much like inserting a key into an ignition, which starts the car. The elongation phase begins with what we call RNA polymerase. This serves to unwind the segment of DNA. Through unwinding the DNA segment, we can clearly see the nucleotide bases. Let's consider the top row, which is the DNA strand, as the strand that will be transcribed or copied. Also, the RNA polymerase will align with a complementary pair of RNA nucleotides to the DNA sequence that needs to be coded. Remember that RNA nucleotides are A, U, C, and G: adenine, uracil, guanine, and cytosine. Remember that A and U will pair together, as will G and C. If there is a T in the DNA sequence, RNA has to match it with an A. The last phase of transcription we call termination, which occurs when we have reached a stop signal noted by the select combination of a few nucleotides. For our example, that is UAA. In essence, what has happened is that our RNA polymerase copied, by matching base pairs of DNA to RNA, a new strand that we can now call mRNA, which stands for messenger RNA. The next component of protein synthesis is known as translation. This process begins with the mRNA sequence that was created from the transcription exiting the cell, where it is released into the cytoplasm. From here, the mRNA sequence attaches to a ribosome. The ribosome eventually opens up and surrounds it, making what we call a ribosomal unit. Now we can see how the ribosomal unit covers the mRNA sequence that we now have. What now attaches to this mRNA and ribosomal unit is tRNA, which stands for transfer RNA. Its purpose is to bring with it specific amino acids or building blocks of protein to the ribosomal unit, based on the sequence of nucleotide bases. Ultimately, this chain of amino acids continues to increase in length, and once a sequence of nucleotide base pairs that are meant to halt the process emerges, this process is complete, making what we can now call a protein.